I would just want to announce everybody can feel free to use their glow sticks during my presentation if you'd like. I mean, it's a pretty hard act to follow. But, you know, anytime you hear something that you like, just wave them in the air. I think I'm just waiting for slides to come up. Oh, there's me. So we've got a great lineup of speakers today to talk to you about the different programs, either with FDA-approved drugs or clinical trials going on for exon skipping. And so I'm going to just do a little introduction. So each speaker doesn't have to do the same kind of introduction about what is exon skipping and how does it work. So just to start at the beginning, Duchenne is caused by mutations in the DMD gene, and then you get a loss of dystrophin protein in muscles. And so to just, you know, build upon that, the gene is your DNA, which sits in the nucleus of your cell, and then that copy stays there kind of protected. And there's another copy of it that gets made, which is called messenger RNA. So when we're talking about exon skipping, we're talking about intervening at this step of the messenger RNA and doing something to it so now you can have a dystrophin protein which is being made, even though you have this mutation. So what is exon skipping, and will it work for me? So exon skipping is trying to get at the root cause of the disease, which is this lack of the dystrophin protein, and restore a dystrophin protein. And the way it's trying to do that is by bypassing or skipping over a particular part of the gene, called an exon, to counteract the problems caused by the dystrophin mutation. And this helps the body create a slightly shorter but still functional dystrophin protein. So it doesn't alter your DNA directly, that copy's still in the nucleus, but it acts on that middle step, the messenger RNA. And this is a therapy which is sort of mutation specific, so it won't work for everyone, it only works for some Duchenne mutations. And if you don't know whether or not it'll work for you, one good general rule is if you have a deletion in the exon adjacent to the exon to be skipped, exon skipping might work for you. And that was a mouthful, so we can look at it in pictures in a minute. But if you don't also know for sure, we can also set up a Cure Duchenne Cares one-on-one meeting. You can talk to your neurologist or a genetic counselor. We can actually help you with this on an individual basis. Okay, so just as an example of how exon skipping works, what is up there in blue are the sort of 79 exons that make up the DMD gene. You can think of them as Lego pieces. And in particular, I'll draw your attention to the shape of the boxes, because that's intentional. And so the boundaries between each one of the exons have to fit together exactly for the sequence from one exon to go right into the next one appropriately. So the code to be read appropriately, this has to fit together exactly. And so you can see from 1 to 79, they all fit together nicely. So what happens now if you have a deletion? What we're looking at on the screen is a deletion in exon 50 as an example. And if you removed exon 50 because you have a deletion in it, now 49 doesn't match up to 51 in the way that it's supposed to. And with that, we say the reading frame is disrupted. And so what exon skipping aims to do is say, okay, can we skip over another exon and now get back in place where they'll fit together in the way that they're supposed to. So if you have a deletion in exon 50, and we gave you a drug like an antisense oligonucleotide that masks exon 51, now you can see 49 matches up to 52 in the way that it's supposed to, and you can actually then, the code, the reading frame of the code is restored, and you would get from 1 all the way through to 79 with only missing those 2 exons in between. And so this strategy, which is, it's sort of like a puzzle, right? it was informed by individuals in a couple different ways. It was informed by individuals who maybe had deletions in things like 50 and 51, and so they still produced some functional dystrophin and had more like a Becker phenotype, as well as some individuals naturally skip exons on their own, and maybe, even though they have Duchenne, they produce like 1 or 2% of dystrophin because their body has figured out how to skip on its own. And so the idea was, can we design drugs that actually maximize that process to restore larger levels of dystrophin? And so this example of skipping exon 50 is just one example. It works if you have multiple exon deletions, which is shown here with an example of if you had a deletion in both exon 49 and 50, you can see exon 48 doesn't fit together with 51 the way it's supposed to. But if you also skip 51, now 48 fits together with 52, the reading frame is restored, and you would have a dystrophin protein, which would be almost complete and only missing the parts that was encoded by the deletion in 49 and 50 and 51, which we've skipped. So if you work through this, if you're someone who likes puzzles, you could work through this on your own. I have a little app on my phone, which you could sort of look through and see, okay, what are the possible ways or strategies that you could put deletions back in frame? And knowing what we know about the prevalence of certain deletions, scientists have come up with this sort of like theoretical framework that it looks like about 80% of individuals have mutations or genotypes which are amenable to skipping one exon to getting them back in frame. And they vary in prevalence with sort of at the top, if you read this at a clock, right? Like at 12 o'clock, exon 51, about 13% of individuals with Duchenne would have a mutation which would be amenable to skipping by exon 51. And you can work your way around to see that they get rarer and rarer subtypes. We have FDA-approved drugs for 45, 51, and 53, so that's the first three most prevalent, and that addresses up to 29% of Duchenne cases, and we'll hear about that. And then we're going to also hear about many other clinical trials that are ongoing, and the goals of the clinical trials are multiple. It's to target additional exons. So we have clinical trials going on in 44 and exon 50. So that collectively would target up to 43% of Duchenne cases. The companies are trying to get better at the exon skipping efficiency, getting more drug into the right cells, getting the skipping to be more, restoring higher levels of dystrophin. And we want to do this not just in the skeletal muscle, but also in the cardiac muscle. So you'll hear about that as well. And some of the programs are trying to make the quality of life of individuals better and do less frequent dosing. The FDA-approved drugs are weekly infusions, but you'll hear that in some of these clinical trials they're moving out to once every 6 weeks, and we're looking at once at every maybe 2 months. And so with that, this is just a little, if you want to take a picture, I see people pulling up their phones, because it's hard to keep track, right, of who's working on what. These are the companies we're going to hear from and about their programs, and I look forward to all of this for all of you. So thank you.
Hi, everyone. My name is Nicole Lamarca, and I am with Patient Affairs at Sarepta Therapeutics. I am so excited to be here today to speak with all of you. Many of you I've known for many, many years, but I've also had the opportunity to meet so many new families this weekend, and it has been absolutely amazing. So I am so privileged to be part of this Duchenne community. I was thinking about it, and I'm almost at 20 years of being part of the Duchenne community. For many years as a nurse practitioner for patients with Duchenne muscular dystrophy, and now with Sarepta Therapeutics. And I have the privilege of being able to really work with families, listen to their stories, learn, understand your hopes and dreams, and I can take that and truly bring it into what we are doing at Sarepta Therapeutics. Your stories are what drive us to keep moving forward to make science better for patients with Duchenne muscular dystrophy. So thank you for having me today. I'm going to be focused today on our exon skipping therapies. These are our forward-looking statements, so you can see them here. All right, so I'm going to speak to you a little bit about all of Sarepta's exon skipping programs today, but first of all, I'd like to talk about our commitment to Duchenne. So we are 100% committed to Duchenne with these 3 technology platforms. So we have our RNA platform, which we'll talk about, our gene therapy, which you heard yesterday from Wendy earlier. She spoke about our approved gene therapy for Duchenne muscular dystrophy, ELEVIDYS, so that was very exciting to hear about. And we continue to research with gene editing. Again, our goal is to be committed to Duchenne and work to find best treatments for patients. All right, so I'm not going to get into too much detail. Liana did a great job explaining what the ramifications of a lack of dystrophin are, but really stressing that at the heart of what we are doing at Sarepta is trying to restore that lack of dystrophin. So we are aiming to target the root cause of the disease at the base level at the bottom to restore that dystrophin, and we are able to do that with exon skipping therapies. I'll go through this one quickly. Again, I think Liana showed a great Lego slide. I like the train analogy. On our website, which we'll show at the end, there's a wonderful video that really explains how exon skipping therapy works. But again, probably the key thing here is the importance of genetic testing, making sure you know your genetic diagnosis to see if you are amenable for one of these exon skipping therapies. So I am so proud and honored to say that Sarepta has 3 FDA-approved exon skipping therapies under the accelerated approval pathway. This is rigorous science that's been done, and we're just so proud to have these products on the market now for patients. So our first therapy for skipping exon 1 is Exondys 51. Then we have for skipping of exon 53, Vyondys 53, and exon 45, Amondys 45. And if you have any questions about these, please, we have a booth, happy to answer questions throughout the day. All right, so this is a busy, colorful slide, and you're probably thinking, are you gonna read through all of these? And I am not. I just have this slide up here because I truly want to show the breadth of research that is going on with Sarepta's PMO data generation. And I think it's really important to know it wasn't just one study, it wasn't just 2, it is a breadth of evidence and that we continue to work on. So not only in phase 1, phase 2, phase 3 trials, but even in the real world. And what that means is just in general clinical practice, how do these drugs function? How do they work? And we're able to really look at things such as how is PMO therapy affecting your breathing? How does it affect your heart function? How does it affect your survival, your walking, something called healthcare resource utilization? So how often are you in the hospital or going to doctor's appointments, things along those lines. So really this huge breadth of data that we're continuing to push forward and really committed to sharing this with the community. So we want to make sure that as we're generating this data, getting this information, that we are transparent in everything that we are doing and making sure our scientific learnings are not only going to healthcare providers, other industry members so they can keep working on things, but also to the community because we know this transparent sharing of data will make us win as one. So continuing to share and grow together as a community. I want to share that with our phase 3 studies. So for Mission and Essence, if you've been to these conferences before, you have heard us talk about these studies, but I'm really proud and excited to say that both Mission and Essence are fully enrolled, and we're expecting them to complete Essence the end of this year, Mission the following year. So we're really excited, and we'll have some more data hopefully in the following years to share with you. And then again, our ongoing study Evolve, which is again real-world evidence where we're looking to see how this drug works for patients in routine clinical practice. So I didn't want to not mention Sarepta Assist, which is our patient support program at Sarepta Therapeutics. It is a fantastic team, and their role is really to work with each and every one of you to make sure that they can help you navigate the healthcare system. We know that there are so many barriers out there trying to access therapies, and this dedicated team is really here to work with you. So if you scan that QR code, you can set up one-on-one appointments or go over to the booth and you can meet with them there. And then I have a few other resources for you here. So for advocacy at Sarepta.com, that takes you to the patient affairs team. So that inbox, it's monitored by all of us here. From patient affairs team, raise your hand if you're in here. That is who will be the ones answering your email. It's not going to an anonymous inbox, and we usually get back to you very, very quickly. And then there's another QR code if you're interested to learn more about any of the 4 approved products that we have at Sarepta. That QR code will take you to our website. So I just wanted to say a giant thank you to Cure Duchenne. This has been an amazing, amazing conference for families, for industry, for healthcare providers, for us to all meet and learn from one another. And we are just especially grateful to the participants and families for participating in our clinical trials. We can't thank you enough, and your participation does not go unnoticed, and none of this would be possible without the commitment of you and your families to drive science forward. So we thank you all so much, and I am out of time, but you can find me in the booth later, so thank you all so much.
Well, howdy, folks. Good morning. My name is Jay Chauhan. I'm from NS Pharma. I'm a medical science liaison. I'm from Houston, Texas, so it was a great drive over here. Today, I'll be discussing Viltepso, taking you, kind of portraying a story for you about Viltepso and how it shaped us as a company to show us, improve our pipeline as well. This is just a reminder that I may be making forward-looking statements during my presentation. So who is Viltepso for? Viltepso is indicated for patients that have a confirmed mutation of the DMD gene that is amenable to exon 53 skipping. Our recommended dosage is 80 milligrams per kilogram, and it is given weekly over 1 hour. As far as safety, I just want to mention that in our clinical studies, no patients experienced kidney toxicity. However, that may be a monitoring parameter that your doctor may monitor for. So this is a great illustration from Cure Duchenne, actually, and this is just showing the percentage of population that we're targeting. You may see in the future pipeline slides showing which exons they're targeting, and I get questions like, why did you start with this one versus this one? And I think initially, as for clinicians and clinical studies, we want to able to capture the most amount of patients in the beginning to prove efficacy. I'm going to take you on a journey through our pivotal phase 2 trial, and this is a story of 16 boys, 8 in a low dose, 8 in a high dose. The high dose was the FDA approved dose, and this was over 24 weeks, muscle biopsy before and after, and what we looked at was, our primary endpoint was looking at how much dystrophin is being produced, and our age range was boys from age 4 to 10 years old, and what we saw after 24 weeks is an average of 6% dystrophin being induced compared to baseline of 0.6%. And I'll also mention that all patients showed an increase in dystrophin from baseline, but the average was 6%. As far as safety, I'm happy to report during this trial, there were no treatment-related adverse serious events, and no discontinuations or deaths occurred. So we completed that study. We got the dystrophin percentage number. Now, how is that going to translate in the real world to skeletal muscle function? I'm happy to announce that during this trial, all patients continued for 4 years, and that's remarkable to get safety and efficacy data over a long-term period. And that's what I'll be discussing as well in this, is looking at our time to stand, which is the primary outcome. And other measures were focused as well, which you may all be familiar with in clinics, such as 6-minute walk test, time to run, walk 10 meters, time to climb, and the North Star as well. So let's look at the time to stand, which is our primary outcome. So over 4 years, you can see patients that were on Viltepso highlighted in the blue line. You can see a change from baseline of 2.7 seconds. And then the natural history group, you can see that separation, which is highlighted in yellow, of 8.3 seconds. And consistent with our safety from our original trial, the first 24 weeks, no participants reported any serious treatment-related adverse events. And this is a further breakdown of adverse events that were reported overall over the 4 years. Now, that trial was pivotal for us as a company, and it allowed us to seek accelerated approval for Viltepso. And we listened to the community. Do you have, data on non-ambulatory? Do you have data on pulmonary outcomes? So went to the drawing board, and we designed a trial, which is called Galactic 53. And that was a study done over 1 year. And that was the basis. This is a story now of 20 boys, 10 that are ambulant, 10 that are non-ambulant. The average age for the ambulant boys was around 10 years old. And the non-ambulant cohort, the average age was around close to 16. And something I'll point out is what we looked at was lung function and motor function as well. In lung function, this is a test that many of you may be familiar with in the clinic. It's FVC. This is usually the gold standard measure in pulmonary outcomes. And it's just forced vital capacity. And what we can see also we measured was, more specifically for the non-ambulatory, is the performance of upper limb. So let's take a second to look at this 2 graphs. On the top is the ambulatory participants, on the bottom is the non-ambulatory. And when we measured their lung function, what we saw at the end of the trial was an average change of 8.3% in the ambulatory cohort on Viltelarsen versus the natural history group 1.2%. And if we focus our attention to the non-ambulatory participants, we actually see a greater separation of 1.6% increase in their FVC from baseline versus a natural decline of minus 3.2%. When we look at the performance of upper limb, what you see is a minimal change from baseline, which is as expected and reassuring as well. Similar with our other study, we were able to see the safety profile of exon skipping, which I'm happy to report no treatment-related serious adverse events or discontinuations. So that brings me to here. Viltepso is an approved product in USA and Japan that really shaped our company and our future pipeline. We have 2 trials that are currently recruiting, exon 44 skipping and exon 50 skipping. I urge you to go to our website or clinical trials or speak with any of us, and we can direct you to find out where the sites are. Furthermore, we have other molecules in the preclinical stage as well. And I'm also excited to announce that we do have a partnership with Capricorn Therapeutics for an investigational cell therapy product for Duchenne muscular dystrophy. Finally, I have to plug in NS support. If you haven't had a chance, please meet us at the booth or find our lovely ladies here that will help you. And this is something that we've established to get one-on-one guidance for you through no matter where you are in your DMD journey. And that's what we're here for. And I'd love for you to meet with one of us one-to-one. And finally, just a thank you to all of you. Honestly, none of this would be possible without your participation, the doctors, the families, the researchers. When I started with NS Pharma, we've grown, and that's due to the involvement and success and investment from the community. And we really value your feedback, how we can design better trials and bring products forward. So once again, thank you all so much. I hope you enjoyed this. If you have any questions, please find any of us. Thank you.
Hi, everyone. My name is Joshua Lilienstein. I'm a medical director at Avidity Biosciences, and I'm here to tell you about the progress that we're making in addressing DMD. Really excited to be here. I hope by the end of this presentation, you'll be as excited about our data as I am. Avidity is based in San Diego, California, but we really bring people together from all over the world to focus on DMD. So today I'm going to talk about our clinical trial for treating DMD, which is amenable to exon 44 skipping. So I'm going to talk to you about an unapproved drug. It's data from a phase 1/2 trial. This drug has not been approved for use, and the safety and efficacy have not been established. So our approach to DMD is by using an antibody oligoconjugate. What does that mean? So it's a molecule that has these three parts: the antibody, the oligo, which does the exon skipping, and the conjugate, the thing that links them together. So let's talk about why that's important and why it matters. The antibodies are these amazing proteins that are made in all of us right now by the immune system. They're part of our immune system. And what's so amazing about antibodies is they're really good at sticking exactly to what they're trying to stick to and not to other things. And so at Avidity, we've engineered an antibody that sticks to the TFR1 receptor. So this is a protein that's on the surface of muscle cells. It's on the surface of muscle cells in the heart as well. And so we use this antibody to deliver the oligo exactly where we want it to go, to the muscle cells and the heart cells. And the concept here is that by doing that, we can hopefully increase the efficacy or how well the drug works, get a better safety profile, and be able to dose less frequently. So that's the concept. So let's see how that's working in real life. We are, as I said, first going after DMD that's amenable to skipping of exon 44. The nickname that we have for that drug before it has a real name is Delzota. And the trial was called the Explore 44 trial. So the Explore 44 trial had two parts: the randomized placebo-controlled trial and an open-label extension. In the randomized placebo-controlled part, we took patients who had, again, DMD amenable to exon 44 skipping, treated them for 7.5 months, followed them. We also had a healthy volunteer part of that study. And the point of that study is really to help us understand what's the right dose to use and what's the safety profile, general tolerability profile of the drug. And then the patients who finished the phase 1/2 study had the option of continuing into the open label extension, in which they're followed for another 2 years. And new patients with DMD 44 had the option of joining the open label extension as well. The enrollment in that open label extension is complete, and both studies is complete, and we're following that open label extension until that 2-year mark. So this is a little more background there. We designed this study in collaboration with experts, with patient advocacy groups, and that's really critical to make sure that we're meeting the needs of the community and really involving all of you in the process. We enrolled in the healthy volunteer study adults. In the DMD part of the study, people ages 7 to 27, ambulatory and non-ambulatory. So we always have to look at safety first, right? In the placebo-controlled part, 2 participants discontinued due to adverse events. 1 due to a serious adverse event of anaphylaxis. It's a kind of allergic reaction that was fully recovered. And another participant discontinued due to an adverse event of an infusion-related reaction, also resolved spontaneously. In the placebo arm, we had 7 patients. In the 5 milligram per kilogram arm, we had 9 patients. And then in the 10 milligram per kilogram arm, we had 10 patients. So the study was really looking at what's the right dose to be using. So now let's go into the, the open label extension. The open label extension enrolled another 38 patients to follow over time. And the safety profile within that part of the study as of January, when we had a last data cut, there had been no serious AEs, no discontinuations. Let's look at efficacy. So how does the drug work? Here we see what happens when we look at dystrophin. On the left, we have what happened to the placebo patients. They really saw no change in dystrophin over the study period. And then we look at what happened to dystrophin in the patients who got either 5 milligrams per kg or 10 milligram per kilo. They both had very substantial increases in dystrophin production, statistically significant of about the same amount, interestingly, in both groups, increased 25% and 26% of normal, up to around 30% of normal. This is a massive increase in dystrophin. So as you all know, that's an important thing, but more importantly, does it work? What effect does it have on how a person is functioning? This study was not designed to really explore the impact of our therapy on functional outcomes. We do have some data on that, and we'll be presenting that at the end of the year. But what we do have is this really interesting biomarker called CK. The CK is very high in young people with DMD because it's a marker of muscle disruption. So as muscles are being damaged, CK is leaking out and is at a very high level in the blood, and we can measure it. So the concept here is that if we are restoring dystrophin to a level that is working for patients, that their CK should come down. So what happens to CK? Here we have a graph of CK. CK decreased profoundly in both treatment groups and stayed high in the placebo group. So in both treatment groups, CK decreased to around 80% from baseline levels, 80% of decreased from baseline levels. And this is a really massive increase, not only did it decrease, but it came down to just around the upper limit of normal, and it stayed there. This is indicating to us that the dystrophin that we're giving back to these patients is doing something. It's protecting those cells from damage, and they're no longer leaking CK out into the bloodstream, and so we're not measuring it. It's really quite low, and that's very different from the patients who got placebo. In the open label extension, the patients who had got placebo previously were able to transition and get the active drug, and you can see that in the shaded part of the graph. So those patients who have very high CK, because they were on placebo, they started getting active drug, and their CKs dropped just like the other people, down to the upper limit of normal. And the same thing happened with the patients who have come into the open label extension primarily. So we've continued to follow all of those patients out to a year. We have even longer data than that now. And CK is staying low. So this is super exciting. I haven't had time to show you all of the data, but I'll just kind of summarize what we've learned here. So we believe that del-zota is associated with the adequate safety and tolerability profile across the doses that we tested. We had equivalent levels of drug seen inside the cell in both 5 milligram per kilogram and 10 milligram per kilogram doses, both doses, and got a really nice, high, consistent level of drug inside skeletal muscle. We had big increases in exon skipping in both dose levels. We had big increases in dystrophin production in both dose levels of around 25% increase, up to around 30% of normal. And we saw huge decreases in CK that were rapid and sustained in both doses. As a result of that, we've decided to move forward with the 5 milligram per kilogram dose, the lower dose, because it really didn't, the profiles of the two doses were the same, and it's always better to expose patients to less drug if you can. So what's next? We're not stopping at exon 44. We have another program recruiting for exon 45 using the same technology. We're really excited about that. The FDA has told us that they are open to receiving an application for accelerated approval, so we're working very hard on that. That should be submitted by the end of the year. We have fast track status, and as I said, we're going to be sharing functional data at the end of this year. We're continuing in DMD. Exon 45 is next. We're looking at other programs as well, and none of that would be possible without contribution and collaboration from patients, from patient advocacy groups like Cure Duchenne. This is a group effort, and we're so happy to have your help. If you'd like to connect, stay in touch, please scan this QR code. You can do it now, you can do it at our booth, and that will allow us to communicate with you and keep you up to date as we progress through our development program and bring new and what we really think are game-changing therapies to DMD. Thanks so much.
All right. Hi, my name is Ash Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. Thank you for having us here. First, we're also joined by Catherine Beaverson, our Head of Patient Affairs, Tiffany Thompson, our Head of Operations, and please meet us at the booth or anywhere else. We'd love to be able to talk to you. Before I begin, I just want to thank Hawken, Liana, Mike, and the entire Cure Duchenne organization for having us. This has been an extraordinary meeting, and then really, I think Yuva and Nate and Ryan should take their show to Vegas. I mean, it's absolutely unbelievable, so thank you for that. It was amazing. So I'm going to talk to you about our investigational program, our investigational medicine, DYNE-251 for Duchenne muscular dystrophy in 51 skip-amenable individuals. These are forward-looking statements, and please refer to our website if you are bored. I'm going to cover 3 things in the next 5 or 6 minutes, and a little bit is a refresher. I've had the privilege and the opportunity of being at this conference for years and speaking to you, and it's always an honor. It truly is. We learn a great deal from you, and we're able to communicate what we're doing and the progress we're making. So I'm going to give a little refresher on our scientific platform called the FORCE platform. You've seen me talk about this a few times. Talk a little bit about our preclinical data. I'll point out a couple of things, and then really get into the clinical data that we released earlier this year that we're really excited about. So Josh did a great job of introducing some important concepts that are overlapping with Dyne's technology as well. We have a FORCE platform that consists of 3 components, the antibody, linker, and payload. We also have an antibody. Here you can see in the Y-shaped figure, it's a monoclonal antibody that binds to that transferrin receptor 1 or TFR1 that you heard about. And why it's important is because it's ubiquitous. It's highly expressed across your muscle cells, including your cardiac muscle, skeletal muscle, breathing muscles, as well as your smooth muscle, your gut. So it's a really elegant way to drive the exon skipping drug into the cells. So we took a portion of that antibody because it's smaller in size. It doesn't have some of the domains associated with immunogenicity. So we took this Fab or antigen binding fragment, engineered it to bind tightly to the transferrin receptor, and attached it with this linker in the burnt orange here that you can see to the exon skipping PMO, our antisense oligonucleotide. And it's versatile. We can attach different kinds of PMOs, 53, 45, or other antisense oligos or proteins or other kinds of payloads to attack the genetic basis of disease. The transferrin receptor, as we mentioned, is highly expressed across the cell membrane. So we have our conjugate that binds to the TFR1. It gets driven into the cell, and then in this endosome, the PMO is cleaved, and then it travels to the nucleus to attack the genetic basis of Duchenne. This is preclinical data that has been published in the past, and I'll just make a couple of points. In this MDX mouse model, which is a mouse model with a 23 mutation, so we used a tool compound amenable for 23 skipping, we saw that when we compared this to delivering the PMO unconjugated or with no delivery system, it barely gets into the diaphragm or the heart or the quadriceps. But when we deliver it with our FORCE platform, you can see robust tissue concentration. So we are getting into the tissues that matter, and actually with a single dose that is sustained over 4 weeks and even 8 weeks. On the right-hand side, you can see dystrophin-positive fibers. It's really important to understand if you're actually able to get dystrophin to the cell membrane. And the green staining on the top row of the right-hand side here shows in the diaphragm and heart and quadriceps, we're able to get the dystrophin to the cell membrane in a robust way. And the bottom part is this, the red staining is laminin, and it just shows that we're able to restore the cellular architecture. And this is really important because those cells are leaky, as you all know, and that's why you have that elevated CK. So this is the trial that is ongoing right now. And we have gone through the MAD portion on the left-hand side, the multiple ascending dose portion. And we are really pleased to announce that we've selected our go-forward dose into our registrational cohort, which is 20 milligrams per kilogram dosed every month. I'll show safety data from all of the cohorts, but I've highlighted the 10 and 20 mg cohorts because that's the efficacy data or the functional improvement data that I'll show you. Very briefly, we looked at, we're evaluating patients who are 4 to 16 years of age, both ambulant and non-ambulant, because we know this is an incredibly underserved group, and looking at safety, tolerability, as well as change in dystrophin from baseline, and many other endpoints too in terms of muscle function, as well as other biomarkers. And everybody now has been escalated to the 20 mg per kg cohort, and those who are in 40 have been de-escalated to the 20 mg per kg cohort. The registrational dose and dose regimen, as I said, has been selected at 20 mg per kg, dosed every month. And I'm really pleased to say that that cohort has been fully enrolled and will have data at the end of the year. These are the baseline characteristics. I'm obviously not going to go into a lot of detail, but I want to point out maybe a couple of nuances. Generally, the patients who are in the 10 mg and the 20 mg per kg cohort are similar. But the 20 mg per kg cohort is a bit more impaired than the 10 mgs. You can see they're about a year and a half older in that first row of age, 8 versus 6.5. And if you look at the NSAA, that fourth line from bottom, you can see that they're at 15 when they came into the study versus the 10 mgs per kg at 25. And they're also a bit slower in terms of 10-meter walk run and an endpoint called stride velocity, 95th centile, which I'll talk about in a moment. So the 20 mg per kg cohort is a bit more impaired, and I just want you to keep that in mind when I show the efficacy data. There's a lot in this slide. Overall, Dyne 251 we believe is safe and tolerable. That is a favorable safety profile. We did see 3 drug-related serious adverse events in 2 participants. I'm pleased to say that both of those individuals are recovered and doing fine. We've had no more drug-related serious adverse events since then, and that occurred in the 40 mg per kg cohort. And as I mentioned, everybody is now at 20 mg per kg. Very importantly, we've seen no persistent related anemia or thrombocytopenia. The reason I bring that up is because the transferrin receptor 1, it's naturally occurring in our body, and its function is to maintain iron transport. So not seeing these effects is really important. We're not disrupting iron transport by using the body's natural cell biology. And no participants have demonstrated any changes in electrolytes like magnesium or potassium. In terms of dystrophin production, we're really pleased that we've seen about 3.7% dystrophin from patients who were treated when you look at baseline to 6 months. It's robust dystrophin expression. And we also do this muscle content adjusted dystrophin here on the right-hand side. And so what we've done is tried to be transparent about how we calculated this. And you can see the calculation there on the right-hand side of the slide. But really what we've done is we've looked at the biopsy sample and we've seen how much muscle versus fibrosis there is in that. And we've then looked at the dystrophin with respect to the muscle, the viable muscle in that biopsy. And we see about 9% dystrophin when we do the muscle content adjusted dystrophin. And that's important because that's going to underpin the functional changes that we see. So this is a measure that you may be less familiar with, but this is really important because it's a real world measure. It's done in the home. And it's correlated with measures you know, like the NSAA and 6-minute walk test. It's also more sensitive, so we should be able to detect changes sooner than some of the other measures. And again, this is complementary to the clinic-based measures that we've also done. Just to orient you to this slide, the gray line is placebo, and you can see baseline to 6 months with placebo. And after 6 months, all patients go into active drug in the open label extension. In the teal, you have the 10 mg per kg cohort dosed every 4 weeks. And you can see that at month 12, all patients who were in 10 mg per kg cohort are now at the 20 mg per kg for that last 6 months between month 12 and 18. And the black line is the 20 mg per kg cohort dosed every 4 weeks. And we're really pleased that we're able to see early and sustained functional improvement in the stride velocity 95C with these 2 cohorts. So we're pleased that we're able to see this real world measure showing functional improvement through 18 months. And it's also above what is clinically important. So it's one thing to see a change, but are you seeing a clinically meaningful change? And we are. This goes through measures that you're also familiar with, the NSAA, time to rise from floor, and 10-meter walk test. And we're seeing long-term functional improvement versus baseline across all of these endpoints through month 18. So in totality, when we look at all of these clinic-based and real-world measures, we're really pleased to be able to see this functional improvement out through 18 months. So I won't go through all of these points. One thing I will highlight is this last bullet, that we have shown preclinical data with respect to the brain and the CNS. I did a webinar on April 1st of 25 that had a video that showed some of this data. And very briefly, we were able to show that our platform delivers the exon skipping drug into the brain, different compartments. It induced exon skipping and produced dystrophin expression. And in fact, we did a standard behavioral model in the MDX mouse that showed we could correct an anxiety phenotype. We know that there's anywhere from 30 to 50% of individuals with Duchenne who have neurobehavioral or neuropsychiatric or neurocognitive issues. And so we're pleased to be able to explore this further as we move into phase 3. And finally, I just want to thank all of you, the participants in our study, the families, the sites and investigators that have dedicated decades of your lives to advancing the science and medicine in Duchenne. It's nothing short of extraordinary, and none of us would be here without you. So thank you so much.
Great, thank you. Thanks, Ash. Hey, good morning. My name is Carla McDonald, and I'm the Chief Corporate Affairs Officer for a biotechnology company called Entrada Therapeutics. There we go. Also a disclaimer, these just basically say in very small print that we're going to share some comments that are forward-looking that will happen in the future. And again, they're available similar to what Ash said on our corporate website. At Entrada, our mission is to treat serious diseases with intracellular therapeutics. And we're proud to receive tremendous amount of feedback from the community as we evolve our programs and as I talked during this presentation, our clinical development activities, materials, and protocols. And we are truly privileged to be able to amplify the stories of several families within your community in our materials, in our websites, and really importantly within our company to help continue to educate all of us at Entrada on the very specific needs and desires of each of you within the Duchenne community. We're a smaller company, and we're a little further behind in the pipeline than the companies you've heard to date. So I put in this slide just to give you a sense of who we are. We were founded in 2016. We're located in Boston. There's about 170 of us. And we spend a lot of time on both really curiosity, exploring deep science, but we also spend a lot of time on understanding culture and the culture of innovation. And we're really proud to not just have a really strong group of PhDs, MDs, and others who are informing our science, but we're really proud to have a really diverse representation of people and backgrounds as part of our company so that we have a better job and do a better job of understanding the uniqueness of experiences. And that is translated into us being a top place to work for many years in a row. And what's important about that, it means that people stay at our company and we have the opportunity for continuation of thought and strategy, and we're really excited about that. And those are just a couple of folks from our labs who I get to work with. I'm going to show you just a little video that talks about our endosomal escape vehicles to give you a bit of a high-level overview of the science. And then after that, I'm going to talk to you about what we've done so far in our first clinical study and what you should expect from us over the upcoming months and years.
If you were to look inside the cells in your body, you'd see a flurry of activity. Every part plays an essential role in keeping the cell and the body healthy. With all these critical functions taking place, it's no surprise that roughly 75% of all disease-causing targets are located inside cells, yet most are still considered inaccessible. There are two major challenges to delivering a therapy to a target within the cell. First, getting the therapy inside the cell. Then, most importantly, avoiding the cell's natural clearing systems, so the therapy can successfully reach its intended intracellular target. Some small molecules can permeate cell membranes, but then tend to be rapidly cleared by the body before they reach the intended tissue. They also may fail to bind effectively to their intended target and can be associated with off-target effects. On the other hand, biological therapeutics like oligonucleotides, enzymes, and antibodies are generally potent and specific, but are limited in their ability to reach their intracellular targets. As a result, high doses are often required to produce a therapeutic effect, which can cause toxicity. Even when a biological therapeutic is successfully internalized, only about 1 to 2% will escape from the endosome to reach its intended target. At Entrada Therapeutics, we have developed a proprietary library of endosomal escape vehicles, or EEVs, with the potential to enable efficient intracellular delivery of highly specific and potent therapies to disease-causing targets previously considered inaccessible. EEV therapeutics consist of two essential parts, an EEV and a therapeutic modality. An EEV is a small peptide from our proprietary library. Its cyclic structure ensures rigidity and stability. The EEV is chemically linked to a specific and potent therapeutic modality, such as an oligonucleotide, enzyme, or antibody. Once inside the endosome, the low pH enables the EEV therapeutic to rapidly and efficiently exit the endosome and enter into the cell's cytosol to access its intracellular target. Unlike biological therapeutics, approximately half of an administered EEV therapeutic can reach its intracellular target. At Entrada Therapeutics, we are driven to transform the lives of patients by establishing EEV therapeutics as a new class of medicines.
Great, so that's a quick overview of the science behind us, and so what are we doing with it in the neuromuscular space? So I've captured our pipeline here, and there's two programs in neuromuscular. One is myotonic dystrophy type 1, and that's being advanced by a company in partnership with us called Vertex. And the other one is our portfolio of exon skipper programs focused at Duchenne. And you'll see in here that they're at different stages in development, and they address people living with Duchenne who are exon 44, 45, 50, and 51 skip amenable. We ran a clinical trial last year in healthy volunteers. So there were 32 healthy adult volunteers, and they received a single IV dose of ENTR-601-44, which is our therapeutic in development for 44 skip amenable. And I just want to take a couple of minutes to share the data with you. The outcome measures, of course, most importantly, are safety. Following, we want to look at how the drug is circulating in the body, as well as its effects on the body. And we're hoping to see some level of target engagement as measured by exon skipping. However, you're not going to see as much exon skipping, or nearly as much as you will with someone who has Duchenne, because these folks have a complete reading frame right now because they don't have Duchenne, and by skipping, we're actually taking them outside their reading frame. So it's a bit of an odd look, but we're just looking for some level of activity. And so the results from this trial were presented last year, and what we saw were no treatment-related adverse events, so no adverse events related to the study drug. There were some events, but they were related to headaches, which were considered mild or moderate, and there were no clinically significant findings in lab values, ECGs, or vital signs. And so we're really, really excited about the promise of the safety of our platform, and that has emboldened us to move this program forward, not just in 44, but 45, 50, and 51. As I mentioned, we were also looking at the pharmacokinetics, so how it's circulating in the body. And we were really pleased to see the strong concentration within blood plasma. And basically what it's doing there is it's supporting the potential for effectiveness at lower doses, because kind of like the video hypothesizes, we're really seeing our ability to get in and circulate within the body. And then finally, I mentioned we looked at target exposure and target engagement. And you'll see here we did do a muscle biopsy. Again, these were healthy adult volunteers, and we saw gosh, that's a tongue teaser today, skeletal dose-dependent reaction in the skeleton muscle cells. And of course, there's a quick illustration of the skipping there. So as I kind of wrap up what to expect, what's next, we've received approval to start multiple ascending dose trials in patients in the UK, EU, and Europe. The UK and EU will be starting this year. In the US, we'll look forward to progress next year. Our 45 programs are advancing again in the EU and Europe this year. We plan to file for our 50 program later this year and 51 next year. And then finally, I just wanted to end on our commitment to the community. We can't do this alone, and so I just wanted to share a couple of ways in which our interactions have directly informed our programs. And your community members have contributed to our study design, our study protocols, our study awareness materials, and also the support materials that we'll be using as we move into clinical development. And then finally, a big shout out to Cure Duchenne. They've got some great statistics demonstrating the number of companies that they have helped support through their financial investments, and we were one of those companies. And so a huge shout out to them for not just running these programs, but also really investing in the science that is really advancing and you're learning more about today, not just from Entrada, but many of the other companies out there. So thank you very much, and good afternoon.
Hi, everyone. Thank you for having us here today. I'm Lee Tai. I'm from Wave Life Sciences, and I want to thank the organizer first for the opportunity to share data with you today. And also, I'm honored to represent our team at Wave Life Sciences. I will be making forward-looking statements today. So, today the first session is really unbeatable, and it was so inspiring, and it really made me realize the importance of grit and resilience and in the community, and it's something at Wave we aspire to. We've been at the DMD drug development for more than 10 years, and really working with you and the community is inspiration to us, and it keeps us going. And we remain very committed to this community. So today, I'd like to tell you a little bit about the phase 2 open label Forward 53 study. This study was designed a couple years ago, really starting with our platform technology, trying to understand what our platform technology can do for DMD. And we have since built on it over time and add more cohorts to kind of understand what this drug we call Wave N531 does. So I'd like to tell you a little bit about that. So first, to summarize, the 48-week data from Forward 53 really show improvement in some of the clinical functional tests that we did. And also, the key thing is that it's distribution to not only muscle fibers, but also some of the stem cells. And we also saw an improvement in CK decrease. And this is supported by the consistent high exon skipping, as well as consistent dystrophin protein production. So this is a little bit about the baseline characteristics of the Forward 53 trial. The trial enrolled patients, 11 patients aged 5 to 11, and all of them have been on stable steroid prior to enrollment and have continued to be during the trial. And consistent with our data from 24 weeks, the safety profile remained favorable with no drug discontinuation. And all of the patients continue to remain on the trial and have continued to receive drug. So this is a slide talking about dystrophin, both in terms of exon skipping and also expression level at protein. So we show that comparing between 24 and 48 weeks, we see consistent exon skipping at around 50%. And this translates into a mean dystrophin expression of about 7.8%. And this is consistent between 24 and 48 weeks. Very importantly, we see that over 85% of the boys achieve dystrophin greater than 5% on average. This is important to have this consistency in a cohort. So what does this mean to have additional dystrophin or new dystrophin produced? This is something we've been following since the beginning of this trial, and really is the focus of my talk today. I'd like to tell you a little bit about muscle health and what we see as a result of treatment by N531, our drug. So in healthy muscle, when there's an injury, we first see inflammatory cells accumulate in the injury site. This is physiological and important because it both causes inflammation but also causes repair. So in a healthy tissue, this inflammation and repair will happen. Stem cell, which regenerates muscle fiber, will be activated, and they will form immature muscle fiber. And we know they're immature by the location of their nuclei. They're localized. The nuclei is the center, the head of the cell in a way. It's localized in the center of the myofiber. As the immature myofiber transition into mature myofiber, the nuclei move to the edge of the fiber. So this is something we can track by looking at histology. In addition, we see a mature muscle fiber shown on the little circle that is very structured and it's correlating with function. In a DMD muscle, when it's damaged, the inflammatory cells also accumulate, but it can accumulate for much longer time and cause more profound inflammation. And the stem cells are activated, but they're impaired and they're not functioning properly. So instead of forming immature muscle cells and then mature muscle fiber with function, it's replaced with fatty tissue and also scar or fibrosis. And this would impair function. So the next few slides are what we have done to track the transformation of the muscle tissue from 24 weeks to 48 weeks. So on this slide, it's a kind of a very typical slide that you will see in histology slides. And there's two things I want to point out. First, the right side, the left side of the slide is the 24-week, and the right side is the 48-week. So comparing the 24-week to the 48-week, you see that there are more purple colored dots in the 24-week, which are inflammatory cells. So there's a decrease of inflammation at week 48. And the other thing is the structure of the muscle fiber. At the week 24, the muscle fibers are more irregular, whereas the muscle fiber on the 48-week looks more like those of the healthy tissue. And this has been scored in a semi-quantitative fashion by our pathologist. And we see that, so this is the result of that scoring. And the higher the number, the more inflammation or cell death on this figure. So you can see that from week 24, which is on the top panel, going to week 48 on the bottom panel, we see a shift from more severe cell death inflammation to less. The other markers that I mentioned are a couple things. One is the centralized nuclei that we saw at week 24. At week 48, we see a decrease of this centralized nuclei, which suggests to us that the nuclei are moving to the edge of the myofiber, which indicate that they're more mature. On the left-hand side of the panel, we see a decrease of the stem cell. Stem cells are activated when there's injury and repair is needed. So having a decrease of stem cell is actually a sign of less need for repair. And very importantly is the amount of fibrosis we saw in these slides. In every single patient that we were able to have tissue to analyze, there's decreased fibrosis from 24 to 48 weeks. Again, consistent with the story that we're seeing of improved muscle health. How does this translate from microscopic to macroscopic parameters? This is a parameter that you're very familiar with, the CK. So we see that there's a decrease of CK from the beginning of the trial to 48 weeks of treatment. And compared to a natural history study, which is the pink line on the top of the right panel, the drug, post-drug treatment has more decrease of the CK. That's expected for patients after a year of treatment. So because this trial is small and also open label with everyone getting treated by N531, we compare our result to a natural history study to look at some of the functional outcomes. So we're happy to find that in time to rise or time to stand assessment, compared to a well-matched natural history study, we saw an improvement by the fact that the natural history is expected to deteriorate in the pink-purple line, but the WAVE N531 treated in blue line is less in terms of the deterioration. We also see positive trend in other assessment as well. So in conclusion, we believe that WAVE N531 can potentially meet some very important needs for boys with exon 53 amenable DMD. And this includes dystrophin expression consistently and evidence of our drug to be able to get into the stem cell. And also we have moved our dosing frequency from every 2 weeks to every 4 weeks based on the half-life calculation from earlier part of the trial. So this will be a monthly dosing regimen. And we're really happy to see the histology slides indicating improved health for muscle fibers. So going forward, the next step, we are continuing to dose all of our patients at a monthly dose and getting more long-term efficacy and safety data. We're also expanding the Forward 53 to including additional boys on a monthly dosing regimen to understand that regimen better. We're planning to engage the FDA to plan a global confirmatory trial, and I would love to come back to share with you the planning and design of that trial when we are ready. And furthermore, we are initiating near-term clinical programs for other exons shown on the slide, and hoping that we can really support more patients with Duchenne muscular dystrophy. So finally, I really want to take this opportunity to thank all of you for your investment in science, in data-driven decisions, and also really for all of the boys, family, and clinician and staff who have helped us in not only for running the trial, but also giving us feedback and advice so that we can know how to best serve this community. Thank you very much.
Thanks. Good morning. My name is Matt Fisher. I'm an Associate Medical Director at BioMarin. And today I'm going to talk to you a little bit about our trial, BMN 351. This is just a forward-looking statement. And today I'm going to be discussing, the first part is going to be looking at the asset itself. So it's an antisense oligonucleotide amenable to exon 51 skipping. And then the second part will be looking at the phase 1/2 trial we're currently recruiting in, and then looking as well at our phase 2b study, which is coming up. So at BioMarin, our purpose is to be the biotech leader that translates the promise of genetic discovery into medicines that make a profound impact on the life of each patient. This is just kind of an overview of our impact. I think for me, the main kind of takeaways are that there are over 14,000 individuals with rare disease currently taking a BioMarin medication. We have 8 commercialized medicines, and we're hoping to launch 11 new medications before 2034. So yeah, more about the study itself, or the drug itself. So as I mentioned, it's an ASO. It's amenable to exon 51 skipping. It's given as an IV infusion, and it's targeting a new site on the dystrophin gene. So basically, BMN 351 is kind of an iteration or an improvement on our first generation molecule, which was drisapersen. And what's different is that there's been chemical modifications designed to improve the stability of the drug within the body and also to increase its capacity to bind to the exon. And as mentioned, it's designed to target a new site on the dystrophin gene, which preclinically has demonstrated kind of increased dystrophin expression. This is just an overview of the inclusion and exclusion criteria for the trial that is actively recruiting at the moment. So the main takeaway is really that it's the boys have to be between 4 and 10, currently ambulatory, and be on steroids for at least 12 weeks beforehand. And then, yeah. And then just a little bit more about the study's overview. So it requires weekly dosing, but we are now kind of moving into home visits. So that has made quite a big difference. I think the health tests are probably pretty standard. And how long will the study last for? So this phase 1 trial is 48 to 56 weeks. And we're predominantly obviously looking at safety, but also interested in efficacy as well. This is an overview of the phase 1 trial. So we've got cohort 1, and that's got 6 participants in it, and that's at 6 mg per kg. This cohort has been fully recruited. And then cohort 2, which is at 9 mg per kg, again, 6 participants, and this has also been fully recruited. And then the third cohort is for 12 mg per kg, and we are actively now recruiting patients into this cohort. So as I mentioned, again, that gives you a kind of an overview of where we're at in the study. Where are the trial sites? So we have sites in the UK, Spain, Italy, Turkey, and the Netherlands. And then just looking what next for the program more broadly. So the next step for us, and we are kind of in the planning stages now of a phase 2b study, which is going to build on the study that I've been talking about, so the phase 1 trial. The duration will be 72 weeks, and it will involve 36 participants, aged between 4 and 21 years. 203 will also be enrolling participants in the US. And unlike 201, so the phase 1 trial, we will be accepting non-ambulatory participants with a Brooke score of less than 4. And then just a little bit about BMN 351, so the asset. So it would likely take longer to reach steady state as a result of the ASO's kind of long half-life. But our hope is that dystrophin levels will increase slowly, but will reach higher levels than other treatments. That's the kind of the ambition. And this will be confirmed through muscle biopsies in the 203 study or the phase 2b trial. This slide here just gives you like the predicted time course of dystrophin expression. And so this is largely based on like our preclinical data and preclinical models. So the y-axis gives you the percentage change in dystrophin, and then on the x-axis it's time. So at 25 weeks, we would hope to see a 2 to 3 fold increase in dystrophin expression at the 25-week kind of muscle biopsy. So just then to kind of summarize the study. So BMN 351 is an antisense oligonucleotide under investigation for the treatment of children with Duchenne amenable to exon 51 skipping. 35201 is the phase 1/2 study, and it's designed to measure basically the safety of the drug, but as I said, we are looking for efficacy as well. And clinical trial sites are open at the moment in Europe, and for the 2b study that's coming up, they will also be open in the US. Thank you very much for your time, and thank you for listening.
All right, everyone, that concludes the exon skipping session. The speakers asked me to rank everybody in order of who did the best job, but I'm going to refrain from making that public. We're ahead of schedule, so we're going to take a short break. But you're going to want to be back here at 11 a.m. because we're going to hear from our champion families, and you'll learn more about their stories of strength and ways that you might be able to get involved too if you're interested. So thank you very much.
Take it easy. your price, just open your eyes. Come right in, take off your disguise. Leave your worries right at the door. Watch them as they drop to the floor. Oh, I wouldn't, have it any other way. I left the room for better days. Your life is sweet like Minute Maid. You shouldn't feel the need to lose your faith. The seasons come and go and change. I just hope you'll hear me say, take it easy, yeah. Make yourself right at home. And never leave me, yeah. You don't have to feel alone. Yeah, oh, oh. Take it easy, yeah. Make yourself right at home. And never leave me, yeah. You don't have to feel alone. Yeah, oh, oh. It's just one call away, and you'll leave him yours loyal to me. But this time I'll let you be, 'cause he seems like he's good for you, and he makes you feel like you should. And all your friends say he's the one. His love for you is true. But does he know you call me when he sleeps? Or does he know the pictures that you keep? Or does he know the reasons that you cry? Or tell me, does he know where your heart lies, where it truly lies? Yeah, you should be with him. I let you go from time. You should stay with him, 'cause he seems like he's good for you, and he makes you feel like you should. And all your friends say he's the one. His love for you is true. But does he know you call me when he sleeps? Or does he know the pictures that you keep? Or does he know the reasons that you cry? Or tell me, does he know where your heart lies, where it truly lies? Right here with me, babe, where it truly lies. My baby, where it truly lies. In my arms, baby, where it truly lies. Where it truly lies. you somewhere, so you know I care. But it's so cold and I don't know where. I brought you daffodils on a pretty string, but they won't flower like they did last spring. And I wanna kiss you, make you feel alright. I'm just so tired to share my nights. I wanna cry and I wanna love, but all my tears have been used up. On another love, all my tears have been used all. On another love, all my tears have been used all. On another love, all my tears have been used all. I wanna take you somewhere, so you know I care. But it's so cold and I don't know where. I brought you daffodils on a pretty string, but they won't flower like they did last spring.